So, our topic today, can psilocybin improve human health span and the biology of aging? So, I'm finding this to be a personally very fascinating topic. I'm really looking forward to this talk. We've got Dr. Louise Hecker joining us from the US and Dr. Macsue Jacques, who is in Australia. And they'll be presenting in the opposite order. So, Macsue will present first and then Louise will join after that. And just before we dive into that, I'll give a little rundown on who we are as Mind Medicine Australia, what our mission is, and a little bit of kind of the context for all of this. So obviously, everything that we're about to say is not medical advice. So, don't try this at home kind of thing. But uh, and we're completely focused on the above ground therapeutic use of psychedelics. So um, we won't be discussing anything in the underground. So the overall context is that we are in the middle of a mental health epidemic in Australia and worldwide. In Australia, we're seeing 1 in 6 people are now on antidepressants, and there's been a 95% increase in the last 15 years. And even in kids, we're seeing 1 in 30 are on some form of psychiatric medication, and this has gone up 196% since 2013. And then in the over 55 demographic, 1 in 4 people are on psychiatric medications. So we've been trying psychiatric medications, the traditional ones, for a long time, and yet the epidemic has been getting worse, unfortunately. And the massive cost really is human suffering. It's the avoidable pain and suffering of human beings. But it does also flow into society in the form of lost human potential, lost human lives in the saddest cases, but also societies, families, and workplaces. This pain can kind of emanate, and we lose a big part of our workforce. We lose. There's a lot of people who are also becoming part of other social problems, homelessness, crime, etc. And in Australia, luckily, we've had pretty big success in becoming the first place in the world to call psychedelics medicine. But it is an ongoing lobbying and legislation field. So, and it's been largely driven by research. So we collect the research, we take that to the legislators, and we go, here's the proof, and what can we do about that if we want to make sure that people survive and thrive? So the traditional approaches haven't really been keeping up with the demand, is one way of putting it. And there's been minimal improvement in the therapies, especially for PTSD, which is notoriously hard to treat. So depression, only 30% of sufferers experience complete remission. And then in PTSD, the remission rate is as low as 10%. So having something new that can become a new approach for people is a big source of hope, especially when you can see how promising some of the evidence is. It doesn't work for everyone, but for a lot of people, it vastly outperforms what we have been able to offer in the past. So Mind Medicine as a charity, non-for-profit, is here for the express purpose of alleviating this suffering through getting people to have greater access to these therapies. And in pursuit of that mission, we've helped to actually get the rules changed in Australia. We were part of that big push in 2023. But we've also created a training organization, which I run, which is the Certificate in Psychedelic Assisted Therapies. And that has trained close to 600 people and quite a few psychiatrists in how to deliver this therapy safely, with efficacy, with the proper amount of care. that is necessary for such a sensitive type of therapy and for such a sensitive demographic. So, and yeah, these therapies are starting to become an integral part of our mental health system, and they're achieving some pretty amazing results. I do this therapy at the Mind Medicine Clinic in Melbourne, and I can personally attest that some of the people we've treated, I've never seen transformations like it. It's amazing. So, very exciting field to be part of. And so, yeah, there's a kind of multi-pronged approach here where we're trying to increase patient access. Uh, we we're a non-for-profit, and any kind of money that we do make, a lot of it goes towards this patient support fund to be able to increase access for people who need the therapy but might not be able to afford it. So if that's you or someone ask your health provider about that. There's a lot of patients that have now received this treatment, and that is thanks to years of work behind that. We now have the clinic in Melbourne, which is the one I work at, and there's also lots of other clinics opening up around the country. We're running these kind of webinars to be able to help people gain information and access. We're doing the professional training. We've got an absolutely incredible faculty of like basically the leaders, the pioneers of this. Like we had last week, we had Bill Riches. Before we had that Professor Nutt. Um, just amazing faculty for this course, and it helps people kind of approach this work with the appropriate amount of training. Then there's research and training as well that we try to provide to be able to help gain the evidence that we need to be able to get this out there. Obviously, in 2023, we had the big change. That was the one where Australia took the lead. And so this is how the news from around the world reported it, which was a refreshing change from the way that it used to be stigmatized. And I think we're all seeing now that it's almost entering the mainstream, you'd say, in the way that so many big voices are talking about how beneficial this is, either for their own lives or for what they're seeing from the research. So we've treated um, more than 180 people. We've got over 50 authorized prescribers. We've got the Patient Support Fund. Medibank has started offering co-payment. We've got a national registry, and we've also got local trials that are going on in anxiety disorder, obsessive compulsive, end-of-life anxiety, and eating disorders. And we also are the biggest supplier of this medicine to clinics around Australia. And we're seeing clinics popping up everywhere. Really nice to see, because obviously getting access to this really helps when it's somewhere near you. So this is the certificate program that we run that gives people the training. It's a 14-week course with a 1 week residential at the last part of it, and it trains people on the delivery of MDMA, psilocybin, and ketamine. We do touch on the other psychedelics, but they're not the focus because they're not the ones that are legislated as potential treatments in Australia. And yes, people can get their CPD points, and if they're a health professional, they can claim it on tax. All that kind of stuff helps as well. Um, if you are a health professional or you know a health professional who would like to take this course, please direct them to our website. Um, and Professor Nutt, who I was just talking about before, is one of the luminaries in this field. Recently said that this was the leading course of its kind in the world. Uh, and I have to say, yeah, from everything I've seen, this is the most comprehensive. So, yeah, a little bit more about the faculty. We've got some of the big names as well. Just to name drop, Gavel Mate, Vesselstep van der Kolk, Rick Doblin. It's an all-star cast. Um, I won't get too in depth with all of them. We also have our fundamentals course. So if you want to learn more about this, but you don't want to commit to the full training program, there is a good Kickstarter called the fundamentals course, which is more like 15 hours rather than 127. And that's another thing to look into if you kind of want to delve into your curiosity in this field. Um, we are a charity, so we do accept help. Any help is always welcome. That can be in the form of volunteering, donating. Obviously, we're always looking for people who would like to actually get out there and do the therapy. Um, yeah, talk to your GP about these therapies. There's all kinds of ways that we can spread the word and that we can build the community. A lot of this is about building the community in Australia, because we have such potential here to do something amazing, and the whole world is really looking to see how we go. So the Patient Support Fund is how people can get access to this, even if they might have trouble affording it. And that offers up to a 50% subsidy in the cost of treatment. And it's been really helpful in helping some people to get access when they otherwise wouldn't have been able to. As that is still one of the major hurdles. So yeah, you can also donate towards other parts of our work, including research and awareness building. We have other events coming up as part of this webinar series and beyond. Uh, so next we have improving the odds of surviving cancer through the mental health code cart. We have lots of others that I won't spruik all of them, but you can see them here, and you can refer back to this if you want to take a screenshot, or just come on our website and we have the events page, which is always being updated. And then one of the big events that's coming up is the Bessel van der Kolk tour, which starts next month. And so he'll be giving some workshops on his work. And anyone who wants to come to that, please come to the website, check it out, sign up. It looks like it's going to be a very exciting event. And that is where I will probably leave our slides and come back to our esteemed guests for today. And I'll start with Macsue and allow you to take the lead.
Okay, so I'm just going to share my screen. Okay, can everybody see this now?
Yeah, that looks great.
Yeah, all right. Okay, so good morning, everybody. My name is Macsue Jacques. I am an aging researcher at Monash University. And, my focus really is understanding aging processes and understanding how can we somehow live healthier and potentially even longer, but really, how can we maintain health? And how does our body or organs age at different rates, and so on. So I look a lot of the molecular changes that happen with aging. And today, I guess, we're going to be talking a little bit about how can psilocybin improve the health span and the biology of aging. So when most people think about psilocybin, and that was very evident by Reese's talk, we think about consciousness, or we think about altered perception, or mystical experiences, or depression trials, or trauma therapy, PTSD. So these are all the things that really come to mind when we're talking about. But really, very few people actually think about all of the other aging processes. So I'm going to be introducing a little bit of a different way to think about psilocybin. So when we talk about aging, we're talking about a multitude of changes that are happening in our body, and some of them are really well known, such as telomere shortening, inflammation. There is also changes about mitochondrial efficiency, but no one has really so far, or has just started, and you're going to hear much more about it later on from Louise, started to link the effects of psilocybin to all of those markers of aging. Because, could this compound, such as this, known for increasing psychological flexibility, also influence biological flexibility? And if it does, what would that mean for aging itself? So let me just take a little bit of a step back to why these questions are only coming up now. So the psilocybin was really first isolated in 1958 by Albert Hofmann, and in the 1960s, early psychiatric research has explored the potential for depression, addiction, and existential distress. So it was always really focused around the mind health or mental health. But then this wave of research really ended abruptly due to regulatory shifts that happened in the 60s. So there was a massive war on drugs that was declared by Nixon back on the day, and all of the research around any psychedelic has been completely shut down. So for nearly 30 years, this has been completely stopped, and only recently in the early 2000s that research really took back into starting trials around those medications. And new studies now have started to show that this promising effects in treatment for resistant depression with long-term non-remission, end of life anxiety in case of cancer patients, also treatment for PTSD, reducing addictions, also help with the OCDs or obsessive patterns of thought. And across all of these studies, people started to say that they feel less rigid, or they are less trapped into those repetitive patterns, and they are more emotionally adaptive. And this is described in research as a restored psychological flexibility that I just mentioned in the end. So the brain in itself, it becomes more plastic. The networks are loosened up and the patterns are reorganized. But here is where things actually pivot. So if flexibility is not just a psychological concept, flexibility can also be a biological one. So how do we start reframing this into looking at the whole entire biology of the body and not necessarily just in the brain? So if you think about aging is often described as not simply the accumulation of damage, such as, as we observe by wrinkles or gray hair or functional decline, but the biology of aging is the progressive loss of plasticity and adaptive capacity across systems. So if psilocybin is doing, really that rescuing of flexibility in the brain, could it be doing in other parts of the body or in the system overall? So when we are young, what happens in our body is that we have the capacity to recover very quickly from infections. We have the repair tissue working very efficiently. We adapt to metabolic stress. We regulate inflammation precisely. So we are really good in responding to any adverse effects. But as we age, what we start seeing is that recovery slows down, the inflammation starts to linger, the mitochondria, which is the molecule inside our cells that produce energy for all daily functions and for our cells to work, they become less efficient in this energy production. And the stress levels start to really become a problem inside our cells. So aging is not just simply the time that is passing, but is actually this gradual stiffening and the loss of this one flexible biological system, and the loss of responsiveness, the less adaptability, and the loss of resilience. So this loss of resilience is what usually drives disease. It's not aging itself, but this reduction in adaptation response. So in recent years, this whole field started to really receive a lot of attention, and that's when scientists started to coin what we call the hallmarks of aging. So the hallmarks of aging are nothing more than markers that are known to be always associated with aging. Initially, there were only 9, but now there's 12 of them. Here, I'm just pointing it out to a few of them that are going to be relevant later on when Louise is presenting her data, which is, telomere attrition. So the telomere basically is the end caps of your chromosomes. And if you think about every time that a cell divides, they're losing little bits of those caps or the ends of these chromosomes. And the shorter they become, the more likely it is that you're going to develop disease, and is also associated with mortality. The mitochondrial dysfunction, as I just mentioned to you, they produce less energy. They are really the powerhouse of the cell, so they become much less efficient in energy production. And this means that there is more oxidative stress being accumulated, which leads to more cellular senescence. So in our body, our cells usually understand that when they lose their function or they are not able to produce the same functions or the same roles that they used to, they should just die. But as time passes, what happens is that some of those cells, they refuse to die, and they become what they call zombie cells or senescent cells. And those cells also release a lot of inflammatory markers, which are termed as SASPs. And this increases the inflammation of the body, which it's a very common term in the aging itself called inflammaging. So the more inflammation we have, the more stress response we're going to have. So you can start appreciating here that those hallmarks of aging, they're not single markers in itself, but one thing really leads to another, and everything is really interconnected. So they are really amplifying each other and just really leading to this whole entire network problem. So that's why aging research is so difficult to treat. One, aging is not necessarily considered a disease, but just, a combination of different things that start going wrong. But we can't really fix aging in itself, or, just fix on health span if we consider one hallmark at a time, but it's a combination. So what happens in the research in itself is that every time that we try to target aging, we usually targeting one specific marker, and it's very hard to find compounds or treatments that are, targeting a multitude of those at once. That's why, single target intervention often fail, because aging is really systemic. So the question that has really changed the direction. So researchers started to sort of notice, okay, if psilocybin increases flexibility in neural networks, could it also engage in pathways involved in biological flexibility? Could it influence in inflammation? Could it influence in stress response systems? And could it influence in metabolic signaling, cellular repair? And that's where the story becomes really interesting. And that's, I believe, where Louise started her research as well. So here is just a very broad introduction of, okay, what are the markers that are being changed with aging? And then now I'm going to pass on to Louise, so she can actually tell you what she found in relation to all of those hallmarks.
Great, thanks. That was a great intro. Let me just pull my slides. Okay, can everybody see my presentation mode?
Mm-hmm. Yes.
Okay. Okay. All right, everybody's good? Great. Couple of disclosures. I have a few companies that I've founded. And so I'm going to tell you really the story of this research, which is, believe it or not, now a comic book, thanks to our friend Chris. So I'm going to use some of his illustrations to tell the story. But it really started with, and I don't study, prior to this project, never studied psychedelics, never studied psilocybin, didn't know anything about it whatsoever. It's really started with a friend of mine who was a neighbor and my workout buddy, and he just kept asking me questions about psilocybin when we were working out. And the scientist in me, wanted to give him the scientific answers, but I just really didn't have any answers. And so, Aaron was getting his information from, social media, and scientists. We get our information from PubMed, which means, I can only look at what's peer-reviewed studies and published, and that's what I can tell you about, what we know scientifically. And so I decided one day to just go read on PubMed, and that way I would have some answers for my friend later on during our workout. And I started reading, and I was instantly intrigued that there were so many clinical studies with psilocybin for so many different disease indications, as we heard about already: anxiety, depression, addiction, OCD, PTSD, bipolar disorder, to neurodegenerative diseases, chronic pain, anorexia, cluster headaches. So that was intriguing to me. Another thing that I just could not really wrap my brain around is that people have reported that just taking one dose of psilocybin leads to positive impact 5 years later. And so for me, wrapping my brain around how is that possible? This has a short half-life. It's pretty much out of your system within 24 hours. So how is that possible? And there are very limited published studies on psilocybin, and in fact, pretty much 99% of what we know about psilocybin either comes from what we know about what it does in the brain or what we know from clinical outcomes. Very few studies have studied anything other than that. And then I came across this one paper that really piqued my interest, called the psilocybin telomere hypothesis. Now, telomeres are the protective caps on the ends of your chromosomes that protect your DNA. And this is a really well-validated marker of biological aging. So as we age, these telomere ends become shorter and shorter until your cells just stop dividing. And so the hypothesis wasn't based on any study as much as a body of data suggesting that positive psychological states are associated with longer telomeres, and negative psychological states like anxiety and depression are associated with shorter telomeres. And so given that we know clinically that psilocybin impacts things like anxiety and depression, maybe it's having some kind of quantifiable impact on telomere length. And so that was kind of the idea. But of course, there were no studies on this at the time. And I thought, well, I'm an aging researcher. I actually know how to study this. But before I jump into the first experiment, I think it's important to talk about the history of psilocybin research, which we've already touched on earlier. But yes, the fact that it became a Schedule I drug in the 70s really created this decline in research. As you can see here, we mapped all of the psilocybin published and funded studies ever in the United States. And it was really surprising to me that there are only 300 studies. I mean, for something that's been in over 200 clinical trials, that seems crazy. To give you a frame of reference, like resveratrol has like 20,000 papers. So, and part of the reason for this is because this Schedule I designation really creates a number of barriers, administrative hurdles for us researchers to study this. And so that led to this kind of sharp decline, which is why we know so little about it right now. But the first experiment was really simple. So when you ingest psilocybin, it's broken down into psilocin, which is the active metabolite. So that's what your cells see systemically in your body, is psilocin. And the first experiment was just using these models of replicative senescence. So cells, just like people, have a natural lifespan, and the experiment was essentially watch these cells over their normal lifespan with and without psilocin treatment. And what we found is that psilocin treatment led to this dramatic extension in lifespan that was dose dependent. So more psilocin, you have more dramatic impact on life extension. And that was sort of the very first finding. And from there, we wanted to look at what's happening inside the cells. So young cells, as they grow old or senescent, they have these kind of very characteristic changes in their cell markers, like increased cell cycle arrest, less proliferation, less efficient DNA synthesis, things like that normally happen accompany with aging. And every single one of these markers was rescued in a dose-dependent manner with psilocin treatment. And this also corresponded with decreased oxidative stress levels. As you can see, at the highest doses, the oxidative stress levels were actually even lower than in young cells. So there's very potent impacts on oxidative stress. We saw that, again, as cells eventually grew old, that process seemed to be delayed. So they had this delayed onset of cellular senescence in cells that were treated. And then again, surprisingly, we found that the telomere length was actually preserved in psilocin-treated cells. So, one of our questions now is to try to uncover how these things are all working together. I mean, do you have less telomere shortening, and that leads to more production of your DNA and less oxidative stress, or do you have more, less oxidative stress, which leads to less telomere shortening, and what's causing what? We're still trying to kind of tease those things out, but the net effect is that you have this quantifiable impact on a number of different hallmarks of aging. Did I stop sharing my slide? Oh, can you still see my slides?
No, I stopped.
Oh, hang on a second. Sorry about that. Clicked something.
Mhm.
There we go. Okay, back. Okay. Oh, why is this not sharing? Oh, okay. Can you see again?
We can see, yeah.
Okay. All right. So based on our, exciting cell data, we decided that we wanted to try this in vivo. And now, considering we only had a small amount of pilot funding to do these studies with, we sort of had one shot on doing this right. And so what we wanted to do is really design an experiment that made sense in terms of that it could be a realistic intervention. So what does that mean? Means we don't want to try an experiment where we're giving mice psilocybin from birth, because that doesn't really translate to something that we would take realistically. When we're seeing these onset of age-related disease, it tends to happen more in your 60s. And so we wanted to start with mice that were the equivalent of a 60-year-old person and see how it impacted their lifespan or their health span. And so we gained insight from not only all of the prior mouse studies that have ever been done, but also clinical studies and trying to design the most effective protocol. And this is what we landed on. We gave an acclimation dose, and then we gave the mice a psychedelic dose once a month. Somebody asked me in the audience of one of my talks recently, were the mice tripping? Yes, they were. We know because they have what's a characteristic head twitch response, which is indicative of that they're having hallucinogenic impacts. So we can see that the mice have the head twitch response, and then it was a bit anticlimactic. The mice went to sleep. But the important part is that now we wanted to do this study and to look at maximal lifespan, but the Animal Care Committee would not allow me to do that study, in part because at the time there was no evidence scientifically, aside from our unpublished studies, that this could even work. There were safety concerns. Psilocybin has never been given repetitively And it's certainly never in the context of aging. And so there were some safety concerns that this study had just never been done before. But so what they did approve was a median survival study. And so when any treatment group got to median survival, which is 50%, that signaled the end of my experiment. And so when the vehicle mice reached 50% mortality from essentially dying from old age, we still had 80% survival in the psilocybin treated mice. Now, again, when we started these experiments, these are the vehicle mice. You can see they're already showing some phenotypic signs of aging. You can see some of the bald spots, and the sort of the hair is a little bit, I guess, messy. And that seems to be exaggerated as they get older. But in the psilocybin treated mice, you can see that actually not only did the psilocybin treatment improve these phenotypes, but these mice at the end of the treatment period actually looked better than they did at the start of the experiment. So white hair grew back in black, bald spots grew back hair. And it was quite exciting for our group to see that physically there was a huge difference that we could visually see. Okay, so the paper came out, which I'm sure is you may have already seen, had a lot of viral attention. So it's this online attention score is apparently in the top 1% of 32 million articles. So we've received a lot of attention. I had no idea that this topic would be so of such interest globally to so many people. But what I want to share next is some of our unpublished studies that are coming out next. We sort of wanted to put the finding out because we were having trouble getting traction, getting funding, getting people to believe these findings. And so without our our publication, we didn't have any, what I call street cred. So we got our paper out, and now I'm going to show you some of the unpublished work that's coming very quickly behind that. So we showed from these mice that we did the study on, we had collected a lot of different organs to assess how, organ-specific impacts of psilocybin treatment. So we found that actually over time that the psilocybin mice had reduced brain atrophy. We gave them some spatial learning memory tests like this water maze test. So essentially the mouse trains where they can see this platform above the water, and they learn where the platform is. And then on day 3, they drop the platform below the water. And so the mice that remember where the platform is swim to that platform quicker. So that's a test of their spatial learning and memory. So psilocybin treated mice had improved spatial learning memory. One of the things that we did that is really exciting is we've taken an omics approach. We had no idea really how this is impacting systemic processes of aging. And so by taking this omics approach, we can sort of look at all the different levels of different organ systems and how they're being impacted from epigenetic, transcriptomic, proteomic, metabolomic. And so we have a whole bioinformatics team that is, working on this data. One of the things so what we've done here in all of our bioinformatics approaches is we're comparing the changes, the transcriptional changes from young to old, or so what normally happens with age. And then we're comparing that to old psilocybin treated mice versus old vehicle treated mice. So essentially what we're looking at in all of these comparisons is what does psilocybin treatment do to bring signaling back toward the way it appeared when it was young. One of the interesting things you can see here is, of course, we expected the brain would be impacted, but it's not even the most impacted organ. The lung had the most differentially expressed genes that were rescued. And then if we look at, again, organ-specific hallmark pathways, one thing that was interesting is that out of the 50 pathways, over half in 4 out of 5 organs were rescued, meaning the signaling is more similar to the way that young mice have signaling. And again, the brain wasn't even the most, had the most rescued pathways, which again means we have so much to learn about what psilocybin is doing to these other organ systems. So again, the lung had the most number of pathways rescued. The kidney sort of consistently we've seen seems to have less protection. We don't know exactly what that means yet, but sort of consistently we're seeing that there's less, I guess, organ-specific protection on the kidney level compared to some of the other organs. One of the big findings that we uncovered is that there seemed to be a really potent anti-inflammatory impact. So as you can see, I've highlighted here all the immune pathways that have been downregulated in response to psilocybin. So we see this massive, doesn't matter where we look in the organ or blood, we see that inflammatory responses is a top pathway, and this corresponds with our proteomic profiling. So inflammatory responses are the top pathway downregulated by psilocybin in the blood. I won't go into details here, but we're doing all this metabolomic profiling, and I didn't know what these metabolites were a few weeks ago, but essentially these are apparently, on a quick search, important for immunomodulatory and neuroprotective effects. So we're starting to gain some insight about, which metabolites are being impacted that are leading to these systemic impacts on aging. One of the other things that we did in parallel is we did a microdose study. So microdosing has been popular. We hear about it a lot. Hasn't been studied as well in clinical trials, but we have some information about it. And the way that we designed this protocol, so different from the prior protocol where we were giving just once a month, mice in the microdose protocol, we're getting a non-psychedelic dose every other week. So they have it in their water bag a week on and a week off. And interestingly, in this study, we didn't find a significant impact in survival, but you could see that there's a trend here. And so we have some ideas behind why this may not have been significant. One being that this is an underpowered study with pilot data. I think anyone who does aging studies is typically would look at these numbers and say this is not enough mice in the study. But also one of the things we didn't think about was tolerance. And I've learned since that when the serotonin receptor is activated, it becomes internalized, and it actually needs a washout period for it to resurface and be fully functional again. And so we think that we may have needed a little more of a break in the off period in order for the microdose to be effective. But that said, microdosing still had some interesting impacts that were different than the high dose. One of the things we noticed was this very striking difference in appearance. So again, one of the big differences is the frequency of this treatment was much different than the prior study, right? They had it more frequently because they have it a week on, a week off. And so we saw these really dramatic impacts in hair and skin that were even more significant than the high dose group that was receiving it once a month. And we also saw that, again, in line with this kind of like hair skin protection, that so normally mice, as they age, they get dermatitis, which is kind of like this dry skin, things that happen with normal aging. And you can see that microdose mice had only 1% occurrence and 0% reoccurrence. So there is some kind of impact that, and what we're learning is that different dosing frequencies and the exact dose are going to be really important in terms of maybe different dosing regimens provide different types of benefit for use systemically. These are just understudied things right now. But certainly, given that we saw this improvement in the hair and skin, we thought, and since we know that wound healing is impaired with aging because of things like increased cell cycle arrest, increased senescence, things that impede the wound healing process and make it less efficient with aging, we know that psilocin treatment can rescue these things. And so we thought maybe there's some impact in wound healing. And so we did a scratch test, which you can see here. It's like basically a confluent monolayer of cells, and then you essentially put a scratch through it and watch the wound closure process. And so we can quantify that. And you can see here that that with psilocin treatment, we had a dramatic improvement on the ability for wound healing. So now psilocybin, as you're all aware, is a Schedule I drug. So it's in the government's eyes the same as heroin. I know in Australia you've had some big pushes to make this, access to patients easier for therapeutic uses. We're still behind you guys in the US on that, but we have identified it's actually a more potent psilocybin derivative that does not cross the blood-brain barrier. And so what does that translate to is that maybe we have an alternative for something that you can take, not have the psychedelic impacts, but still have the positive impacts systemically on aging. And so that's the hope. But again, we still don't know how the mind-body access works in this context. So it'll be really interesting to test. Do you need that mind-body connection in order to experience these positive impacts systemically on aging? So yeah, this is just showing that we've shown all the same phenotypes using this derivative. But really, the big question is, does psilocybin impact human aging? Because if it doesn't impact human aging, then why are we here? And so one of the things that we did is that we can look up disease pathways, human disease pathways, using our omics data. And there's a human disease phenotype ontology database. And we're able to look to see in the mice, at least in the psilocybin treated mice, how many of these sort of disease phenotypes are rescued. And as you can see here, a number of, at least our transcriptomic data suggests that a number of these disease pathways are rescued, and with, again, more organ-specific impacts. Some organs seem to have more protection than others. And so here's, you can see these are all the organs and 4 out of 5 of these organs, all of these things were rescued. A lot of these, again, have to do with inflammatory responses. So it's sort of consistent with our top, inflammatory responses being a major impacted system. One of my colleagues at Emory, they just got a $20 million award. One arm of that award will be to test psilocybin for the first time in healthy older patients. And so it's actually a safety study. And so one of my grants is actually funding an ancillary study along with their study. So I'll be able to actually be collecting the endpoints of these patients and doing all the biological characterization. And then with that, the very first clinical trial for psilocybin and aging will be initiated in 2026 as well. I'm a part of that study where we're also going to be doing similar types of phenotyping. So it'll be really interesting to see in humans how these findings play out. And the last thing I'll mention is that Ali John is on a PBS documentary that, if you look it up, called Your Fantastic Mind, it actually takes you through a patient in one of these clinical trials and his journey. And it's just an unbelievable story. If you have time to watch it and you're interested in this topic, I would highly recommend it. It's just incredible to watch this patient go through this process. So yeah, and I guess I'll just end on kind of a final note, which is that I've spent sort of the whole of my independent career focused on trying to target age-associated diseases. Here's my target of interest. This target goes awry in aging, and this leads to this disease. And I'm still working on that strategy. But this project has really started to make me think differently about what is the best bang for our buck in terms of, age-associated diseases. And I think actually this might be a better strategy overall, is to really think about not getting to this point. If we can just have people live healthier longer, then we can delay the onset and or improve age-associated disease outcomes. That would be better than waiting until you get disease and trying to cure one disease at a time. So that's sort of my new way of thinking about this. And for me, the holy grail is, can psilocybin, we know so little about it, can it be used as a geroprotective agent that can improve health span? And that's what I'm hoping to find next. We'll see. Thank you.
Wow. Thank you so much. What a great presentation.
Thank you. Of
Um, do you have time for some questions?
course, yeah.
Okay. Um, if anyone wants to throw a question in the chat, or you can use, there's a react function at the bottom of Zoom where you can raise your hand, and then we can get to you that way if you want to ask verbally. Um,
I can start with a question while people get prepared or type in,
yeah.
since I'm here. Fantastic talk, Louise. I noticed that a lot of, the pathways that psilocybin is actually rescuing with aging are similar pathways that exercise, for example, rescues as well. And I'm just wondering, have you been able to compare, the effects of, like, let's say, exercise versus psilocybin, or even if potentially the mice after psilocybin became more active, if you actually looked at that? Because they're just feeling better and they're exercising more. So just a little bit curious about that.
Yeah, great questions. I didn't look at that in the study because I didn't want to like subject mice that are in a life extension study to like, other interventions of any sort in case that had any confounding impacts on my study. So the short answer is I don't know. And let me see, there was another part I was going to answer. But it's okay. Yeah, that's what I was going to say. It's really tough actually for me to make direct comparisons to, let's say, like calorie restriction or exercise or rapamycin, because unless you're really designing the study to be a head-to-head study, like when I went back in the literature and I was like, well, how does this look next to calorie restriction? Well, they might have started at this age and my study started at this age, and they're doing calorie. How do you compare something that you're doing every day for years versus you're taking 12 doses or 10 doses total? So it's really hard for me to make those comparisons unless I'm testing these interventions in the same population of mice at the same start time, the same end time. But it'll be exciting because I'm sure that people will eventually reproduce these in the context of those studies, and it'll be interesting to see how it compares.
I'm wondering what the equivalent human dose is. Um,
Yes. That's a tough one because yeah, I've gotten a lot of questions about dosing and how I decided the dosing protocol. And, it was again, keep in mind, so with so few studies, I didn't have
mhm.
a lot to work with. So like I started with like what doses have been shown in mice to be psychedelic versus not psychedelic, because we wanted to stick in a range that we know. Then I was thinking about clinical trials and what's being used, but then I'm also factoring in like, well, mouse metabolism is 4 times faster than humans. So it was, you know, is it like exactly equivalent to what they're being given right now? Not exactly, but it's a hallucinogenic dose. It's meant to model like that what we're doing clinically, which is infrequent hallucinogenic doses.
Hmm.
But yes, more needs to be done on dosing in order to really translate this as a geroprotective agent for the general population.
Thank you. Um, we have someone asking how they can follow your future publications. What's the best way to access
Yeah,
that?
sure. You can follow me on LinkedIn. I always post them on LinkedIn when we have new papers, Louise Hecker, and on Twitter or X. Certainly, looking me up, you can always see my papers as they come out. And just a Google search, I'm sure you'll find it.
And we've got a anecdote of someone finding ketamine to be quite effective in a similar way. Are you aware of any evidence for ketamine in
Yeah,
this
yeah.
field?
I mean, yeah, sure. I think like in light of our findings, we have we have like all kinds of drugs that we're testing in the lab right now. We're testing DMT and LSD and because it's just kind of like, our finding really kind of kicked open the door to, hey, this is, when you think of these drugs, you think of course, the brain. And I know I think of serotonin, I think of the brain as well. But I was really shocked to find that serotonin receptors are on almost every cell in your body. And so it makes sense, right, that it's actually doing something systemically to these other cells that have the same receptor that it works on. Um, but the reality is we just know so little. I mean, there's just almost nothing known about what any of these drugs do systemically because the focus has always been clinical outcomes or what's happening in the brain. And so I think that's why this study is so important because it really kicked down that door. People are starting to ask different questions and look at different drugs in those contexts. And so I think it'll be really exciting to see what happens, not just from our lab, but from all over the world in the next year on, to build on these findings with other compounds.
Yeah. There's another question here about inflammation. Are you aware of the mechanism by which psilocybin lessens inflammation or how it happens?
Great questions. The short answer is no. We're doing a lot of studies with collaborators where we're doing the tedious things of like blocking certain receptors and then seeing the effect is there or not. And so until we kind of do that finish those rigorous studies, we won't have answers. But we have some ideas and we're following up. And we have, one of the great things that came out of having this finding go viral is like people contacting us and saying, hey, we have this tool to look at telomerase activity. And so, or we have these knockout cell lines, or and so we've been able to gain a lot of resources from collaborators that are helping to answer these questions.
Mm. Always new tools coming through. Awesome. Um, so another question here is about stress and considering that the mice might have been less stressed afterwards, and if that was a potential mechanism.
Yeah, I mean, I think anything could be, there's like anything's a possibility right now, sure. Um, yeah, I didn't know. And again, like part of this it was like, well, I don't even know how to measure stress in mice. I'm sure there are ways, but now that we know like this, how important this finding is, the next time we run these studies, like looking at different dosing and things, we're gonna have all kinds of ways to measure things like stress. So one of the things we're doing now to help get a better understanding is actually like take a step back and look at systems like Drosophila and C. elegans, where they have a very rapid life cycle. And in those model systems, you can get all kinds of information, like, for example, like egg laying rates. That's like a measure of, reproductive health. And so we can test dozens and dozens of protocols and look at, and with the Drosophila, which are little flies, you can do this test and you just kind of let the bottle kind of shake and all the flies go to the bottom, and then you can actually measure how fast they crawl up to the top, and that's a measure of motility, right? So we can test lots of protocols to have a better idea of dose, frequency, how it's impacting all types of things, and then kind of bring these protocols back to mice after we have some insight, and further validate them versus, doing those studies all in mice would take, millions and millions of dollars and many, many years where we can have answers quicker that we can then validate. So we're looking into all these things, but we didn't have answers right now.
Given everything that you've researched and seen, how likely do you think it is that that the same effects wouldn't happen for humans.
Yeah, I mean, I'm biased, right? Because it's my research. So of course I believe in it. Um, you know, I mean, I would bet my life these are going to be reproduced in mice, because I've done the studies and I've seen the results. Translating to humans, I'm extremely hopeful. And I just think as a scientist, the only thing I can say is, we'll see when the data's here, but I wouldn't be doing it if I didn't think it was worth doing.
Mhm. And uh, we've got something here about the anecdotal evidence of people who've been taking it for years, such as Ram Dass. Um, do we have any data there that we can draw on?
It's so tough because I mean, I think as a human being, of course, I want to speculate and I want to believe things and I want to understand. But as a scientist, I can't, right? Because we're trained to do rigorous scientific studies, and I'm trained to rely on that and only that. And so I think the anecdotal stories are good in that they can help, just like my friend is asking me questions to pique curiosity and to ask the questions. But I can only rest on what we show scientifically and hold my, and hold my hand on that until we have rigorous studies. I can't really have an opinion about anecdotal stories.
That's completely fair. Bit of a trick question. Back to the science then. How many months or doses was the macro dose experiment for?
Yeah, it was 10 months, so 10 total doses, 1 month apart, which I don't know, maybe roughly is equivalent to about once a year for humans.
Hmm. Okay.
Reese, and there is a
Um,
question here, if I may.
yeah.
Um, how does psilocybin compare to other anti-aging molecules, I'll call it?
Well, again, it's like comparing like apples and oranges unless you're doing a head-to-head study. But I will say that I've seen, other scientists I know, like I've never met Matt Kaberlin, but he's a key opinion leader in the longevity space. And he posted a video where he did a 30-minute breakdown of the figures in our paper. Very
Hmm.
candid. I mean, he tells you what he doesn't like in the study too and what the limitations are. But he does overlay the rapamycin data. And again, the difference being that like his study went for the entire duration of lifespan, whereas mine had to end at 50%. But his data overlaid perfectly with ours. So it looks like certainly it had it's performing well against rapamycin. To know like specifically, I think we have to figure, which is better. We'd have to, like I said, think about dosing, optimal dosing, and head-to-head studies. But yeah, I think seeing other scientists overlay data like rapamycin and saying, yeah, there's something here is really hopeful and exciting for me.
And then, if I may pose a question, um, taking your research, what's the end game? Is the hope
Yeah.
being that a person may just take a tablet every single day and it will help them
Mhm.
with their longevity process?
Yeah, that's a great question. Well, I think, the end game for me, and again, like to me, the holy grail of all of this is can you find a dosing regimen that makes sense, that gives you protection in your later years of life so that
Yeah.
you have an improved health span? Are we trying to live forever? No. And the data suggests in the cells, they're not going to live forever, right? They do have a normal lifespan. It's just longer,
Yeah.
and they'll say healthier longer, and it looks like the same for the mice. And so to me, that's kind of the end goal, is like, what does that look like? And does it have to be, psychedelic? Do you need the psychedelic impacts to have the maximal systemic impacts? That we don't know yet. We're going to have to test. but maybe you can take it and have protection without those. That might be something that's more exciting for a lot of patients that don't want to have that psychedelic experience. So I think that's the end game for me is, can you use this to delay some of these age-associated diseases longer and live healthier longer? And so the jury's still out. We think it can. The data suggests it can, and we just need some more rigorous testing to find out. And along with that, we have to look at the potential harms. I mean, no one, again, like I said, no one's ever given psilocybin, as far as I know, in a human study or animal study, repetitive over a long period of time, and definitely not in the context of aging. So we have no idea if there are any detrimental impacts of this kind of treatment, this long-term treatment. It's never been tested. So things like that have to be tested as well to make sure that there are not risks to this type of treatment.
Fantastic.
And
I just had a request to, I'm supposed to like Macsue, so I'm going to put both Louise and Macsue centered, and hopefully we can get some questions across to both.
and if I may, I see that you said that there was research getting done on other psychedelics and other substances. Are we talking about psilocybin today because that's the one that's showing the most promising results, or is just because it's the one that has the most kind of complete data set at the moment?
Yeah, well, I guess, ours is the first study in history to show in mice that there's a dramatic impact in systemic aging processes and improved survival, even when given late in life. So, and that, just came out 6 months ago. So I think, since our study, a couple of studies have come out, and I know there's something with, I'm not sure if
There's
it's even public.
ketamine as well.
Yeah,
There's
so
some,
things are
yeah.
starting to pop up, but this was the first, and it was a comprehensive in that it cells in mice. So I don't know what these other studies show, but this is all brand new frontier.
So just to complete on that, so there's some publications as well showing that in cells and also in some animal trials, ketamine has similar effects. I haven't compared specifically how much and if they tested the same thing, but again, related to aging. So,
Which jurisdictions globally do you see as the most progressive or least restrictive in supporting the research with a view to bringing it to widespread clinical use?
Well, you guys seem much more liberal than the US right now.
yeah, I was gonna say, I think Australia is one of the first ones to actually have, a lot of the clinics for the general population, while, in the US, you still have to be enrolled in a trial to be able to access any of the drugs. So definitely here's a good place um, to have access to the treatment. And then, in saying that, I guess also puts us in a very good position to run new trials, since, there are people already taking this as part of treatments. And some places also, like in Europe, so maybe, the Netherlands, I know there's a lot of research happening in there in this area. And I think
Yeah,
Netherlands.
in the US, there's 2 states that have, approved it for medical uses, and there's a growing number of cities where it's been decriminalized, but it's still federally illegal. So it's a problem.
I also think it's worth clarifying, Louise, that hopefully your work will lead to a consumer product, so to speak, where a person can have this dosing regime, as you called it earlier, to hopefully slow down their aging. Whereas in Australia, our focus has been entirely, treating chronically mental ill health conditions like PTSD and treatment-resistant depression. And that clinical context is different to the work that you're doing. So hopefully your work will also lead to a more widespread use in a healthy way.
Yeah, I think, I mean, in my personal opinion, I think it's just a matter of time before it gets rescheduled. Um, I mean, it's already in phase 3 clinical trials here. So I think, when there's an accepted medical use, and so it'll be, things will change, but yeah, I think they really need to change. And I wish they would change faster, so I can't wait for that to happen. I'm hopeful that things will get better and better over time, but sure, I think research like this, is needed to destigmatize it. This is, like I was saying, I think before we started, this is an incredible drug that we know so little about, and we need to study it more.
Yeah.
I can't help but think of the prevention is better than cure mantra, and how that is starting to relate more to like this biological aging stuff as well as the prevention of mental health conditions. Um, yeah. Okay, we're getting pretty close to the end. So if anyone has any last questions, please fire them through.
I did
We've got
see
one.
a question here. I'm just going to jump in.
Yeah, go.
Yeah, psilocybin used for treating in combination with other modalities like, dance, art making, and, things like that. There's other researchers actually now that are looking into electronic music, like raving. So there's like the longevity raving movement coming up where they are actually seeing the person that started is also a very prominent name in the field. Her name is Tina Woods, and she started raving after 60, and she noticed a massive decrease in her biological age. And that was literally the only thing that she has done differently from, she already was healthy, exercising, and all of that. And one of the arms of the study that she's now trying to expand is to have this combination. What does just the music do, and what does the music with the combination of the psychedelic use, if it's psilocybin or ketamine, actually changes. So there is more research there coming up in this area, which is really exciting.
There's another one that's just come through about treatment of dementia or other neurodegenerative diseases. Are you aware of anything there?
Yeah, I mean, it's in clinical trials for those things already. I mean, I'm, very hope. I think, one of the things that we have to think about is like, well, what if it's showing all this promise? Why aren't these things happening faster? And here's the problem: is that psilocybin is a natural product, and so Pfizer isn't excited about developing psilocybin, right? It's a natural for their business. And so a lot of these trials and things are being privately funded. And so that's great, but there's not a lot of private funding available relative to federal funding for trials. And so it's all relative, right? It's all relative to the funds that are being fueled into seeing this develop. And if there's no company that's directly going to stand to benefit, then how are we going to fund the trial? So these are some of the problems, I think, that translate into why aren't these things panning out faster?
Thank you very much. I think that's bringing us close to the end of our time. So I want to thank our presenters again for sharing their knowledge, their experience, their expertise. Um, does anyone have any last things they'd like to say? Louise, Macsue,
I may
Alan?
suggest that tomorrow we'll send out a follow-up note. So as part of that, we'll include a special discount code to attend Best Sylvander Cost workshops in Australia. But also, if Louise and Macsue want to send me links to their research papers, it's well worth sharing that with the audience as well.
Great. Yeah, thanks for having us. And, it's an exciting topic. So keep your eyes peeled for new stuff that's coming out.
Fantastic. This is a really good session. Thank you, everybody.
Thank you.
Thanks,
Thank
everyone.
you.
Thanks for everyone for your questions as well and for showing up and being part of this.